Hello, everyone, and welcome to ACS webinars, connecting you with the best and brightest minds in chemistry, live from Washington, D.C. I'm Michael David, and I am pleased to be your host for today's broadcast, which is being co-produced by CAS, a specialist in scientific information solutions. The COVID-19 pandemic has driven an unprecedented surge in vaccine research and accelerated development of new vaccine technologies. Today, we are joined by Dr. Shane Crotty of the La Jolla Institute of Immunology and Dr. Yizhou Dong, who works at The Ohio State University's College of Pharmacy, for a discussion on the progress of the COVID-19 vaccine research, including its potential for future applications for other disease and therapeutics. We would love to get you involved in the discussion today, and you can share your thoughts and questions in the questions panel. You can also join the discussion on Twitter by including @Americhem Society in your tweet. The slides for today's webinar can be accessed right now by clicking on the link you see in the GoToWebinar control panel. An invitation email to view the unedited recording from today's presentation will be sent to all registrants. And now a little word about today's moderator. Today's moderator is Angela Zhou, an information scientist at CAS. She earned her PhD in molecular pharmacology and toxicology from the University of Southern California. She was further trained as a cell biologist at Johns Hopkins School of Medicine and worked in the area of cancer biology at Harvard Medical School. She was also a faculty member at Missouri State University and Denison University prior to joining CAS. And with that, Angela, I will turn it over to you to get us started today.
Thank you, Mike. Welcome, everyone, and thank you for joining us for today's ACS and CAS co-produced special webinar, COVID-19 Vaccine Update, Emerging Questions and Future Application. As Mike introduced me, this is Angela Zhou from CAS. CAS is a division of the American Chemical Society. We specialize in scientific information solutions. We organize, analyze, and uncover connections within the world's published scientific information to deliver actionable insights that accelerate discovery. Hundreds of scientists at CAS take pride in helping researchers in commercial, government, and academic institutions worldwide. Our unparalleled content collection, specialized technologies, and unmatched human expertise can help you maximize the value of scientific information and advance your work. Since the very beginning of this pandemic, CAS scientists have been supporting COVID-19 research and creating resources including journal articles, scientific reports, data sets, and other educational materials to support the global fight against this disease. If you're interested, you're more than welcome to visit our website at cas.org/COVID-19. All those resources are available as open access materials on the CAS website. And a few examples are shown here, including one of the white paper called COVID-19 Vaccine, which will be published soon, either today or in the next few days on CAS website. Now, without further ado, I'm gonna introduce you today's guest speakers. We are thrilled to have two prestigious vaccine experts with us to talk about the most recent update of COVID-19 vaccines and also answer as many questions as possible from you about the vaccines and the related technology. Let's welcome Dr. Shane Crotty. Dr. Shane Crotty is a professor from Center for Infectious Disease and Vaccine Research at the La Jolla Institute for Immunology. His research team studied immunity against infectious diseases and the formation of immune memory. He is a member of La Jolla Institute Coronavirus Force Task. His team's work on COVID-19 immunity was referred by Dr. Fauci as exceedingly important to the field of immunogen design. Thank you so much, Shane, and welcome here.
Thanks for having me.
Thank you. Together, we also have Dr. Yizhou Dong. Dr. Dong is an associate professor in the Division of Pharmaceutics and Pharmacology from the Ohio State University. His research focuses on the design and the development of biotechnology platforms for the treatment of genetic disorders, infectious diseases, and cancers. Dr. Dong has authored over 70 papers and patents. Several of his inventions have been licensed and are currently under development as drug candidates for clinical trials. Thank you so much, Yijo, and welcome here as well. Okay, so Shane, I think I'm gonna start with you, and Yijo, I will turn to my question to you just in a few minutes, okay? My first question, Shane, to you is, up till now, I think there are millions of people have received these COVID mRNA vaccines, but people are still wondering if these vaccines are safe, and people may still need more information about how these mRNA vaccine works in our body, and how does our immune system respond to them. Could you give us more information about that?
Definitely, Angela. And it is, these are new vaccines and relatively new technology, and so it's definitely natural for people to have questions about it. And in fact, I put together a summary that I posted on Twitter of some of the most common questions, and that's shown here. So on the next slide. Really, the first key thing for people to understand is that RNA is messages. People were concerned about the use of the word genetic for RNA, but actually RNA is messages. And so in any given cell in your body, there are currently 5,000 different RNAs, and they really are messages. They're like Post-it notes, each with a different instruction written on them, telling the cell what to do and what to make. And they're all temporary messages, like Post-it notes. They get shredded up and thrown away on a regular basis. And so the RNA vaccines work on a similar concept. On the next slide, if you prefer a different kind of analogy, RNA is like Snapchat, where it's literally a message that expires. It's there for a period of time and then gone. They don't become a permanent part of your body. They're there for a couple days probably. Next, please. In the case of these COVID vaccines that are RNA vaccines, some people have also asked me, well, can that create a new virus, or cause infections? And the answer is absolutely not. They're completely safe in that regard. Coronaviruses are actually fairly complicated viruses, and it takes, as shown here, 25 different coronavirus proteins to make a virus. And in the vaccine, there's just one of those 25, the spike protein. So there's no virus as part of the vaccine. It's just one message. Next, please. And in terms of safety, people have asked, well, how do you know that the vaccine is safe? And that's really the point of the clinical trials. And so then people have asked, well, how much information do you need, right, to know that the vaccines are safe? And in fact, the FDA, the US FDA, asked exactly that of its scientific advisory board. And so they went and looked at all the vaccines that have been licensed to date and asked, how much safety information do you need to know that a vaccine is safe? And their answer was 2 months worth of safety data on several thousand people. And so that was the requirement that the FDA has held any vaccine trial, any COVID-19 vaccine trial in the US to. And actually what's been phenomenal is that in the interest of speed, these have actually been some of the largest vaccine trials ever done, with the Moderna trial having 30,000 people, the Pfizer having close to 40,000, the Novavax aiming for something in the 40,000 range, and the J&J and a similar thing. Those so because they were made to be large trials so that they could be fast in terms of determining efficacy. The more people in the clinical trial, the more cases of COVID-19 you'll see faster. But as a bonus to that, you actually get a huge amount more safety data because there's so many more people who are immunized. And so this slide is actually from the time of the clinical trials themselves, when there had been 70,000 doses given, and all of those people got tracked, right, very thoroughly for any side effects, and they were fine. And so 70,000 doses worth of safety data is a massive data set. And now here we are in February with the latest numbers are 65 million doses have been given. And so there's an incredible amount of safety data, and these RNA vaccines are impressively safe. Next slide. But people do get confused about the word safe when it comes to vaccines. Safe does not mean the same thing as doesn't hurt at all or no fever. And I'm an immunologist, I study the immune system. And one of the things that's important to understand about the immune system is the immune system is very good at ignoring things that aren't trouble, that aren't a threat. And so one of the big successes of the vaccine field over the years has been figuring out how to make vaccines that stimulate the immune system, right? That trigger the immune system to think of something as a potential threat, while at the same time being safe. And so actually, when you have an immune response that you notice to one of these vaccines, your arm getting sore, having a bit of a fever, that's really not unlike going to the gym and getting exercise and getting really sore, right? And you get really sore and you go, oh, well, that's actually a good thing, right? I'm going to be stronger tomorrow. It's a similar thing with the immune system. It can really be a bit of pain can be a positive sign that good things are happening. So this is, sometimes you have to earn your immunity, just like you earn those biceps. Next slide. Which is the same thing. So next, and this is a more scientific schematic of how an RNA vaccine works. This one was drawn by Eric Topol and published in the journal Cell recently. So you get immunized, and the whole point of a vaccine is it needs to be showing your body at least a piece of the virus so that your immune system can learn how to recognize that virus. And so the clever approach of RNA vaccines is using an RNA message to express the viral spike protein. And so you can see that down near the bottom, the viral spikes then get expressed on the surface of cells. And your immune system then recognizes those viral spikes as foreign and learns how to recognize them. And makes a large number of B cells and antibodies and T cells to recognize those and hopefully protect you in the future. Next, please. And so this is the actual clinical trial data, and these vaccines, both the Pfizer vaccine and the Moderna vaccine, worked so incredibly well. The Moderna curve looks virtually identical. That it's actually very easy clinical trial data to interpret. That the blue line going up is the number of cases of COVID-19 in the placebo group, right? So the people who got immunized with just salt water instead of a vaccine, whereas the vaccinated people who actually received the RNA vaccines are the red line. And even within a couple weeks after the first immunization, there was clearly an effect. And so by the end, you can see this 95% protection effect from the vaccine in the clinical trial, and real world data are supporting those findings as well. Next slide. And the final big safety question that I hear from people and having, when I get asked a bunch on Twitter and whatnot, is really in the category of, yeah, but what about people like me? So were people like me in the vaccine trial? Do we really know the vaccine is going to work on people like me? And in fact, these were probably the most inclusive vaccine trials ever, both the Pfizer and the Moderna one, with a very large fraction of people who are over the age of 65, who are clearly the biggest group of people who need protection, but are sometimes not included in clinical trials, right, because of their age. In this case, there were lots of people. It was lots of men and women. Very good representation of the Black population and very good representation of Latino populations. And looking at any of those subgroups, the vaccine's just as good. The RNA vaccines are just as good in all of them, which is really impressive, particularly in the elderly population. So yeah, these vaccines do work in you. And we could talk about pregnancy later if you want. And I think, yeah, I was gonna pause there. Angela, did that answer your question, Angela?
Yeah, definitely. I think it's good news to know that these mRNA vaccines are safe. And like you said, no pain, no gain, right? We need to make some kind of scene for our immune system to recognize this is a potential enemy may come into our body. Great. And I'm also monitoring the news. Looks like from the news, these vaccines are doing the right job protecting people from the disease, right, in the real world. Yeah. So, Shen, I have another question. I actually have heard quite a lot about immune memory, recently, and I know your team focused on this particular aspect. Could you give us more information about how these immune memories could be formed or how long they will last?
Definitely. So that's immune memory is a specialty of my lab, and it's definitely a major topic for vaccines. And we just published a big study in Science on this topic for COVID-19. All vaccines work on the basis of immune memory, and all immunity from infections you've had in the past work on the basis of immune memory. That is to say, your immune system will remember infections you've had or vaccines you've received, and that memory is built into antibodies or cells of your immune system. And so in the slide, the immune system's complicated, and there are at least 4 different components of immune memory. So there are antibodies, which most people are familiar with. There are also memory B cells, which is the purple cell there. So those are cells that can make antibodies. They can make the same antibodies if stimulated. But they're normally quiescent. They're normally resting cells, not actively making antibody. They're the backup manufacturing plants of antibodies. And then you've got 2 kinds of T cells. You've got CD4 T cells and CD8 T cells. And CD4 T cells can do a variety of functions, either having antiviral activity or helping B cells make more antibodies, or helping CD8 T cells, or wound healing. They can do all kinds of different things. They're quite complicated. And then CD8 T cells will directly kill infected cells. The simplest way to think about these parts of the immune system are that antibodies are really good at stopping a virus before you get infected, sort of stopping the virus at the front door. But once you're infected, now the virus is inside of cells, and antibodies can't get to the virus inside of cells. Again, that's an oversimplification, but largely true. Whereas once the virus is inside of cells, you really need these CD8 T cells to be able to survey and go find those infected cells and kill them. So you can have different parts of your immune system really working together, like different branches of the military that can sort of coordinate to fight, enemies in different places. So we wanted to measure all of that as a global measurement of immune memory in people who had gotten COVID-19, to ask, how long does your body remember this infection? And so this study was 188 people across a range of disease severities, and we measured all of these cell types, which is actually the largest such study ever done for any viral infection. And what we found, on the next slide, I'll just show you some raw data just to give you a sense of the types of things we were looking at. We measured, on the left side, we measured several different kinds of antibody responses from our team. And then on the right side, we measured memory B cells, CD8 T cells, and CD4 T cells against the virus using a variety of techniques. And then on the slide after this, and we did it over 8 months. And that was a key thing, which is it's actually quite hard to predict immune memory. We don't have good predictors of it, and the best way to measure immune memory is actually to wait 6 months and let things settle down some and see how much does your body still remember. And so we made sure to wait at least 6 months for the study, and actually it went out as far as 8 months. And we measured memory in different people at different times across that, so we could look at the kinetics of it. And then we combined all that together in the next slide. And basically on the bottom, where it says GB48A, we took the 5 components of immune memory that we thought would be most relevant for protection, and we asked, okay, if we just take a look at everybody 1 month after they're infected, how much of that memory do they have? And then what do they have out at 6 months? And by looking at the 6-month time point, we concluded that 95% of people had at least 3 of those 5 parts of immune memory at 6 months, and they were following the kinetics sort of shown in that previous slide. There wasn't some sudden drop off, there was just kind of a slow decline. And so we've interpreted that as a good sign that most people have a pretty substantial amount of immune memory after they've had a case of COVID-19, which would be likely to protect them, at least from serious illness, for a substantial period of time. And studies like this are now going on for the vaccines as well. But it's definitely possible for your body to make substantial immune memory to this virus and make a number of different kinds of immune memory against this virus, which has generally been a positive sign.
Great, great. Thank you. I am always fascinated about the immunologists. There's so much, we don't know. There's so much to study. I think the work you've done is phenomenal. It's not just laying the, fundamental structure for COVID-19. It may show the future directions, how we should study our immune response to other viral infections, right?
Yeah, that's exactly right, Angela. I mean, when we measured all of these parameters in these people for this study, the immediate question people asked us was for any of the things that we met, any of the different parts of memory that we measured, people said, well, what's that look like for other viruses? And for most of those, we said,
we don't know.
there's really not that much information. This is actually the largest such study for any virus. And so we could tell, like, for example, for the CD8 T cells, it actually looked pretty similar to what the yellow fever virus vaccine gives. And so from that, we can say, okay, this is actually a pretty good T cell response because it looks similar to what a vaccine got. But for some of the other parameters, these are actually probably going to now become major reference points for other diseases.
Yeah, great. So Shay, I'm always, puzzled as well with the COVID-19 disease, that different people respond very differently. Some people are asymptomatic, some people have very severe problems. So do you have any, information about these?
Yes, and so that's on this slide. It's a really important question, Angela, and there's no proof at this point as to why different people are having different outcomes. But there's a lot of data, and there are some reasonable possibilities, and our working model is really shown here, which we developed in September, which is in the upper right. A perfect example of an immune response to a generic viral infection would be you make a very rapid innate immune response within a couple hours, and that's the part of your immune system that recognizes broad patterns of seeing a virus or a bacteria, just in general, can tell that there's some sort of infection. And then that gets followed up later by the adaptive immunity. And the adaptive immunity is what I was talking about before. That's your antibodies and your T cells that can really specifically recognize exactly this virus, okay, and wipe it out. And that's what vaccines do, is generate those very precise responses. But those responses take time. They take a week or more to develop because they're starting from very few cells. And so it is in fact a race. When you get infected, it's frequently a race between the virus and your immune system. So what happens with a SARS-2 infection, a coronavirus infection? That's in the bottom left. So in an average coronavirus infection, with most viral infections, the virus has to have at least one immune evasion mechanism to be able to make people sick. If the virus couldn't evade the immune system in some way, it probably couldn't make anybody sick. And so understanding that immune evasion trick of the virus is frequently important to understand the pathogenesis of the virus. And in the case of SARS-2, it's clear that one big trick of this virus is that it is unusually stealthy at avoiding triggering that early innate immune response. The early innate immune system has trouble seeing this virus. This virus is very silent. And you can think of that early innate immune response as both blunting the viral infection, but also setting off the alarm bells. It's like the fire alarm, right, that something's going on. And so that delay in the early innate immune response, which is predominantly a type 1 interferon response, that gives this virus a head start and allows the virus to grow because the innate immune system isn't holding it back. But it also turns out that your adaptive immune response depends on those innate immune alarm bells going off first. So if the innate immune response is delayed, then your antibody and T cell response is also delayed usually. And so a lot of what happens in COVID-19 is probably the downstream consequences of that one big early event after infection. And the way it works out is that in an average infection, some people, their innate immune response responds so fast and so well, plus their T cell and antibody response being so good that they don't have any symptoms at all, right? Those are your asymptomatic people, which are probably like 30% of cases. Whereas in most people have some symptomatic infection, so the virus got enough of a head start to end up causing symptoms, but then the T cell and antibody response still happen quickly and you're okay. But in a severe infection, our simple model is that basically the same things are happening, but that initial innate recognition is even later and slower. And so this has been shown really clearly in people who have innate immune system deficiencies. They have a really high risk of hospitalization or death with COVID-19. Yeah. And so then those people, it looks like their T cell responses are particularly late. And this also connects to age. So we've observed that older individuals, people over the age of 65, have more trouble making a T cell response fast enough to control the infection. And so we think a lot of this ties into those couple of parameters actually, and just combining those couple of parameters can tell you this wide spectrum of disease, which I'll end with saying, we interpreted all of that as actually good news for vaccine development, because the brilliance of vaccines is they can get you that immune response before you're infected. So if the problem is the race between the virus and your immune system, a vaccine can solve that problem, whether you're elderly or et cetera. And in fact, the successes of the vaccine are consistent with this model.
Yeah.
I'll stop there, Angela.
Yeah. So it looks like the vaccine can set you up early on the start line, right? So you can catch up the race faster. Yeah. Great. Well, Thank you so much, Shane. Yeah, that's great information. I think we must have received a lot of questions from audience, but I will turn those questions to you later. I'm gonna turn my focus to Dr. Dawn. Hi, Yijo, how are you doing?
Hey, Angela, how are you? Thanks for having me.
Yeah, so I know you and your team are heavily involved in developing mRNA vaccine and the lipid nanoparticle technologies. Could you give us, some information about how lipid nanoparticle technology is, and particularly such as what are the compositions, how they are made, or how do they package and deliver nucleic acids such as mRNAs?
Sure. Scientists have been working on lipid nanoparticles for over 5 decades. Different types of lipid nanoparticles have been constructed based on specific therapeutic agents, such as small molecular drugs, DNA, small interfering RNA, and messenger RNA. Regarding the COVID-19 mRNA vaccines, so here's the ingredients of the vaccine. So the active agent is modified. It's a chemically modified mRNA encoding the SARS-CoV-2 spike protein. So that's the active agent. And there are several lipid components to formulate the nanoparticle. One is an ionizable lipid. Others are cholesterol, phospholipid, and PEG lipid, serving as helper lipids. And here are two structures. One is the ionizable lipid used in the BNT162 Pfizer-BioNTech vaccine, and the other ionizable lipid is SM102 used in the Moderna vaccine. So both have similar structure. They have tertiary amine that can be easily positive charged under formulation conditions. These lipids can interact with negatively charged messenger RNA, and the other helper lipids, they can help stabilize the nanoparticle and then form the quite homogeneous nanoparticles. And after the formulation, they also have a component called cryoprotectant. So here in this case, it's sucrose, which is a sugar molecule. It can protect the nanoparticle under low temperature and maintain the integrity of the nanoparticles. So these are the ingredients of the nanoparticle. Next slide, please. And this is an illustration to formulate these lipid nanoparticles. And I borrowed some information from the FDA advisory committee meetings with Moderna. This is their presentation. And during the formulation, they first dissolve the mRNA in water phase, and they dissolve other lipids, including ionizable lipid and helper lipids, into the ethanol phase. And through this, we call that a microfluidic device. The general principle is enable fast mixing of these two different phases, and then they self-assemble into quite nice nanoparticles. You can see here, it's from electronic microscope. The particle size is between 80 nanometer to 100 nanometers. Next slide, please. And this is also the slide from Moderna's presentation. I think Shane already gave a very nice description about the safety. And here, I think they presented the similar message. They only delivered a few copies of messenger RNA into cells compared with over 200,000 copies of mRNA molecules in cells. And also, there is a very minimum or low chance messenger RNA can be reversely transcribed into DNA based on our current knowledge. And so the take-home message is that they think this mRNA vaccine cannot alter our human DNA. So that's the formulation of the current COVID-19 mRNA vaccine.
Thank you so much, Yijo. That's great information for me to learn as well. We've heard lipid nanoparticles a lot, but we don't really know what's the composition there. Great information. Now, my second question is, we know that this technology of lipid nanoparticles may have existed for a while, right? It's not a brand new thing. Maybe people have been explored in many other areas, particularly in drug delivery area. Could you give us some examples how this technology has been applied in existing treatment or maybe for future, therapeutics?
Sure. So in addition to viral vaccines, messenger RNA has been applied to many different types of therapeutic indications, especially in preclinical studies. And also currently there are more pipelines in clinical trials, such as protein replacement therapy, cancer immunotherapy, and more recently, gene editing. So I will share with you a few examples from our research projects. And here, this slide shows the overall strategy our lab tried to develop and optimize lipid nanoparticles for mRNA delivery. And we first synthesize different types of lipids or lipid derivatives, and then we can formulate with other formulation components, as I mentioned, for the COVID-19 mRNA vaccine. And here, the therapeutic agent is messenger RNA. Through the microfluidic-based device, they self-assemble into very nice nanoparticles. Then we can characterize the particle properties. And then we utilize the orthogonal experiment design, which can enable us to efficiently identify the optimal formulation with significantly reduced experiment load. And after that, we identify the optimal formulation, and then we can test it in different disease models. Next slide, please. So here's an example, hemophilia. Hemophilia is a genetic disorder caused by missing or mutation of factor 8 or factor 9. Patients suffer from life-threatening bleeding or serious complications such as joint bleeds. Next slide, please. So in this study, we formulated one material called TT3 with mRNA encoding human factor 9, and then we injected intravenously in factor 9 knockout mice, which is a hemophilia B mouse model. And then we measured the factor 9 activity. So in this figure, the y-axis, that's the factor 9 activity in MIU per ml, and the x-axis, that's a different treatment groups. You can see it showed a dose-dependent recovery of the factor 9 activity. And at the dose of 1.1 milligram per kilogram, the activity is around 800 MIU per ml. In normal serum, the factor 9 activity is between the range of 500 to 1500 MIU per ml. So that means this formulation is able to fully recover the human factor 9 activity to the normal range in these canine knockout mice. Next slide, please. And more recently, we also worked together with the groups of Ron Weiss and Daryl Irvine, and we formulated TT3 with self-replicating RNA encoding IL-12 for cancer immunotherapy. And this is an illustration of the mechanism. We formulated the nanoparticles, and we did intratumoral injection, and then it triggers the immunogenic cell death, and then trigger the subsequent anti-tumor immunity. Next slide, please. And our collaborators did many studies, and here are some examples. So they performed a single injection of the formulation into multiple types of the tumor models in mice. And they found that they can significantly extend the mouse survival in B16F10 model, CD26 model, and also humor 1.7 model. They also performed a series of maximum action studies, and they found here's one example with the knockout of the MID88 or STING, the therapeutic efficacy is significantly compromised. Next slide, please. Meanwhile, we did a lot of chemical modification of these lipids, and we can install different type of the lipid chains, including the carbon chain, branched ester chain, and the linear ester chains. And then we can use these new materials to formulate lipid nanoparticles. Next slide, please. Here's one result to utilize these lipid nanoparticles for base editing. We worked together with Beam Therapeutics. We formulated these nanoparticles with mRNA encoding base editor and guide RNA targeting PCSK9. And on the left, you can see it showed dose-dependent base editing. And on the right, this is a targeted deep sequencing at the dose of 0.125 milligram per kilogram. It still induced over 50% base editing. So to our knowledge, this is the most efficient delivery system reported in the literature. Next slide, please. And lastly, I would like to give you an example. We are thinking to utilize lipid nanoparticles for treating deadly disease, such as sepsis. Sepsis is a life-threatening condition caused by severe infections. This disease affects over 30 million people worldwide and remains the #1 cause of death. And the mortality rate is pretty high, around 25 to 30%. So next slide, please. And this is an illustration of our strategy. We designed vitamin-derived lipid nanoparticles to deliver messenger RNA encoding antimicrobial components into macrophages. And then we use these macrophages to treat septic mice in order to improve the innate immunity, prevent the bacterial immune evasion, and eventually eliminate the multi-drug resistant bacteria. So next slide, please. And here are some structures of the vitamin-derived lipids. We utilize the vitamin B3, vitamin C, VD, VE, and VH. Next slide, please. We performed a series of optimization, and then we identified the vitamin C lipid nanoparticles significantly improved the mRNA delivery in macrophages. And then here's the example. The C5 formulation showed over 70-folds better than lipofectamine 3000 and over 300-folds better than electroporation, which are state-of-the-art for transfection in macrophages. And on the right, that's the TM images. It showed the particle morphology. Next slide, please. And here we established a sepsis model by infecting mice with multi-drug resistant bacteria. Here is a mixed drug resistant bacteria, S. aureus and E. coli. We would like to mimic the mixed infection in the sepsis patients. And on the left, you can see the engineered macrophages significantly reduce the bacterial load in these septic mice. And on the right, more importantly, these engineered macrophages dramatically extended mouse survival. These mice eventually fully recovered. Next slide, please. Yeah, here are some body condition results. These mice continue to grow, and on the left, that's body weight. And on the right, these are the white blood cells and the lymphocytes. You can see all these survivor mice, their white blood cells and lymphocytes return back to the normal level. So here are some examples that we have done, tried to explore the broad applications of mRNA-based therapeutics.
Well, thank you so much, Yizhou. You give us really wonderful, background information of how these lipid nanoparticles have been explored and applied in multiple areas. Looks like this is a pretty well-established technology, and there are a lot of fine-tunes we can do to control these lipid nanoparticles, right? Yeah, thank you so much altogether, Yizhou and Shen, for your wonderful background information. I think at this time, we're gonna turn to our audience questions. We have received a flood of questions, and I'm doing my best job to combine their questions if they, ask the similar aspects. Now, the most frequently asked question is about the new viral mutations. There are California variants. People ask, how would the inoculated people, means the vaccinated people, deal with these new viral mutations? Will they still have enough immunity to go against this new virus? And looking to the future, how frequently are we going to receive an update of these coronavirus vaccines, in order for us to cope the mutation speed? Maybe Shane, you can go first, and Yizhou, you can join afterwards.
They're really important questions. They're big questions, and the answers are shifting every week as we worldwide. People collect more data. I'd say that the quick versions are there are now substantial vaccine clinical trial data, so direct evidence, direct proof for protective efficacy of several vaccines against the two biggest variants of concern, the UK variant B117 and the South Africa variant B1351, with several vaccines having maintain almost their full protective efficacy against the UK variant, and losing some efficacy but still having efficacy against the South Africa variant, specifically the Novavax and J&J vaccine have evidence of that. And just yesterday, there was data, there's been data coming out of Israel about RNA vaccine success against B117 by inference, and that's looked good. And then there have been more direct data from the UK, the Siren study and a Scottish study, which both came out yesterday, that both indicate that the Pfizer RNA vaccine is having substantial protective efficacy against the B117 variant, and the expectation would be that Moderna would do the same. How they're protecting is unknown. So it's as I was saying at the beginning, you've got antibodies, memory B cells, CD4 T cells, and CD8 T cells, and it's unclear what combination is important for protecting against variants and whether all the vaccines are doing the same thing. To me, it seemed like the most likely scenario is that it's a combination of several of those. That your best case scenario is if you have great antibodies that work perfectly and you just stop the virus at the front door. But if you don't have that, having CD4 T cells, CD8 T cells, or memory B cells can be a good backup plan if you have them, because the viruses are exhibiting, some of these variants are exhibiting some escape from antibodies, but they're very unlikely to be escaping the T cell response at all. So whatever the T cell response is contributing to protective immunity is going to be there. I'll try and answer your last question very quickly as well, which is how often are we going to have to get vaccinated? Honestly, the simple answer I give people is that it's basically the same answer as in November, December, which is it's reasonably possible that annual vaccinations will be normal for a while, for either of two reasons. Well, the one is that we just don't know how much immune memory RNA vaccines generate. There's just never been a licensed RNA vaccine before, and as I was saying about immune memory, there's no simple way to predict immune memory. It may be that they provide immunity for a year, but you have to get a booster immunization at a year just to maintain what you had, or that the variants shift enough that you want an updated vaccine that better matches the variant. And I and a lot of people have been recommending that the South Africa variant is different enough that it makes sense to make an updated vaccine that largely reflects that. And Moderna and Pfizer have both said they're already doing that. So there's a good chance there'll be an annual booster for either of those two reasons, at least for a couple of years. I'll pause there and see if Dr. Dong wants to add to that.
That's very informative. I don't think I have anything to add.
Okay, we're okay. Yeah, since there are a lot of questions, I'm gonna jump into the second question. The second question is about the allergic reaction with these mRNA vaccines, and people think this is mainly caused by the pegylated lipids. Maybe Dr. Dong, you will have more information about these lipid modification with PEG. Are there any other alternatives that we may be able to, update the vaccines that people will have less allergic reactions? Could you give us some information on that aspect?
Sure. I think for the current formulation, this pegylated lipid that's already used in previously approved lipid nanoparticle formulations for siRNA-based therapeutics. But for that indication, there's much less patients injected with this type of lipid nanoparticles-based formulation. PEG definitely, there's clinical results showing which can induce a certain level of the allergy reactions, but it's still not 100% sure right now because it's a formulation. There are many components in this formulation, and the previous clinical results are mainly based on usually a single pegylated agent, either PEG conjugated with a small molecular or PEG conjugated with a single RNA. But this formulation is different from previous agents studied in clinical studies. So I would assume there need a lot of more careful studies if exactly it's a PEG or it's a whole formulation or it's a combination with the PEG and the formulation other components. Regarding if there's any other like alternative materials, I think there are a lot of studies ongoing in preclinical studies because this PEG lipid has been used previously in several different type of formulations. It's still relatively safe compared with other materials not tested yet in human clinical trials. So currently, I think other materials are in the preclinical study stage, and I would assume in the future there will be more and more other materials that can replace current PEG lipid.
Okay, great. Thank you. Yeah. The third question is about what happens to the vaccines after they inject into our body. Like most vaccines is intramuscular injection, so presumably it's hitting our muscle cells. How do these mRNA get to our immune cells? And a follow-up question is, will these lipid nanoparticles pass the blood-brain barrier, go into our neural cells and do something in our brain? So any information on that perspective?
On the first one, it's a great question. So your summary is accurate. It definitely gets delivered to muscle cells and expressed on muscle cells. I think it's still unclear whether delivery to other cells that are more immunological cells may be more important. One way or the other, those proteins then have to be delivered into lymph nodes, which are where the B cells of your body, the cells that are actually going to make the antibody, that's where they have to see the spike protein. And some of those steps actually still need to be figured out better. but it's known that similar things happen for other vaccines actually, right? So it's intramuscular immunization is the norm for most vaccines, and yet those materials then get drained or trafficked or carried to the local lymph nodes, which is where the key immune response actually happens.
So do we have any idea about whether they can cross blood-brain barrier?
The blood-brain barrier is very hard to cross, so I would think that would be surprising, but I don't have specific information on it.
Mike, could you unmute Dr. Dong?
I'm sorry, Dr. Dong, you appear to be self-muted, so you might have to unmute yourself.
It doesn't. Oh.
Yeah, you're good.
Okay, great. So I just add a couple of points. I think based on our studies, we found that the lipid nanoparticles injected through muscle tissue, the majority of the particles or expression of the protein would stay at the injection site. There's a very minimum delivery to other organs, and for brain, I think there's even less or even no delivery to brain. I think that's a big barrier, blood-brain barrier, and there's a lot of studies that try to overcome that. I would assume there's a very little chance these nanoparticles can be transported to the brain tissue. And for the degradation, I think these particles after injection, as Dr. Crotty mentioned, the mRNA is vastly degraded by the cells, and the other components, there's several helper lipids that's natural moleculars, like cholesterol and phospholipids. Those are natural molecular, cells know how to deal with them, and the ionizable lipid is also a small molecular. I think that's also be degraded through like esterase or some other hydrolysis process. Eventually will be cleared by kidney.
So mainly their local reactions don't travel much far, right? Yeah.
Right, that's our understanding right now. Yeah.
Yeah. Well, my next question is for Dr. Crotty. This is about the cross reaction or cross protection of immunity between COVID-19 and other diseases. In your research, have you seen any cross reactivity of immunity between COVID-19 and common cold or pneumonia or tuberculosis?
Yeah, so this goes back to, so my lab and Alex Studies lab published the first paper looking at virus-specific immune responses to COVID-19, looking at CD4 T cell, CD8 T cell, and antibody responses, and that paper got a lot of attention. And one, because we were showing that people actually make a good immune response to the infection normally. And so that was actually a reference point that people have used for a lot of their vaccine studies and whatnot. But one component of that paper was that we reported that we could actually detect T cells in about 50% of people who were uninfected people. About 50% of them could already recognize SARS-CoV-2 with their T cells, at least some. And that was surprising and indicated cross-reactivity, which we speculated was coming from common cold coronaviruses. And so there's been a lot of activity on that topic in the past 9-10 months now. And essentially, it's still an unknown. So the options are, one, the fact that some people already have some memory T cells that can recognize the virus may make it so those people are more protected because they already have a head start on their immune response to the virus. And we know that happens with flu. So if with the H1N1 pandemic in 2009, it turned out if you had memory T cells from decades ago, you were unlikely to get very sick because you already had some cross-reactive responses. But for coronavirus, it's also possible that those T cells, the T cells that we measured, they're just not cross-reactive enough to do something useful. Immunology is complicated. The presence of the cells isn't enough information. And it's even possible that those cross-reactive cells are programmed incorrectly, and so would actually have a negative effect on outcomes. And so actually all three of those possibilities are still at play. There's been one big epidemiological study that looked like there's at least a little bit of protection that people can get from those T cells, but it's at the level of, it's not at the level of infection, it's at the level of like hospitalized disease. But we're still waiting for clear studies on that. One of the problems with answering that kind of question in immunology is really the way to answer it is you have to go collect a whole lot of blood samples from a whole bunch of people who aren't infected so that you know what kind of T cells they had before they were infected. Then you have to wait for some of them to get infected, and then you have to compare before and after. And so that takes a lot of time, and that's where we are right now.
Yeah, great. Thank you. My next question is about the transmission. In those people who have received 2 doses of vaccine, and is the transmission of the virus still possible? In other words, are the vaccines protecting from transmission?
So transmission is definitely still possible because nothing's ever absolute in the immune system. So if I always tell people, if somebody tells you something happens 100% of the time in the immune system, you know, they're either lying to you, to keep it simple, you know, or they don't know, because the human immune system is really designed to not always do exactly the same thing in every single person, right? However, I mean, what the question's actually asking, right, is transmission reduced, I think, and reduced how much. There's a wide variety of opinions on this topic at the moment. I will say the best data, the data that are available are in the Moderna clinical trial. They happen to have a piece of the trial B that when people came in for their second immunization, they got tested for virus just as part of that visit, okay? And when that happened, there were cases. They were all asymptomatic cases. But when they looked at the placebo versus the vaccinated group, the vaccinated group had about a 2/3 protection, something like a 66% protection, even after one immunization, against having any detectable virus. And so that would definitely be preventing transmission. If there's not enough virus to be detected in a PCR test, there's no way you're transmitting. That was a small data set, and it wasn't really designed for that. So lots of people have said it's still an open question.
Still open question.
J&J just reported to the FDA in their like 118-page document this morning, so I haven't read every word of it yet, but they reported something similar, that they were also seeing a reduction in PCR positive cases, a significant reduction in PCR positive cases, suggesting that you'll have a certain amount of protection from, basically with these vaccines that are working, you're going to have a lot of protection against hospitalization level disease. You're going to have somewhat less protection against any symptomatic disease, and then somewhat less protection from, is there any virus at all in your nose that might possibly be transmitted? But it looks like, at least for the RNA vaccines and the J&J vaccine, that there is a significant amount of protection from transmission, I think. But it's not proven at this point.
Okay, great. Yeah, thank you so much. Now it looks like we're close to the end of our webinar, but I would like to ask Shane and Yijo, if you would like, us to have a take-home message from today's webinar, what would you tell to the audience? If you can summarize that in half a minute, that would be great. I can start with Yijo.
Sure. I think we have a lot of chemists in this audience. I think chemistry already and will play a critical role in developing new type of materials. And also now it's really a great time to work on RNA-based therapeutics or vaccines. That's it.
Thank you.
Right, with there being a lot of chemists in the audience, I would certainly say immunology is really complicated, but it is really cool and important, and there are lots of things chemistry can contribute to understanding immunology or having, immune system related therapeutics. So please don't be too terrified of all the acronyms in immunology. It is pretty interesting. I would end with saying there are now 6 successful COVID-19 vaccines. That's incredible. There's never been anything like that in human history. Even in December, right, having 1 successful vaccine within a single calendar year had never been accomplished before in human history, and it was quickly 3. So, and these vaccines work incredibly well. So they really are phenomenal successes and incredibly safe. But you know, vaccines only work if you take them. So it's now, I think a lot of the science has really been dealt with, and now it becomes a lot of communication to try and communicate to people that these are safe and effective vaccines, and really you and your neighbors have to actually take them if we want people to really be safe.
Great. Thank you so much. Thank you, Dr. Don, Dr. Crotty. It was a true pleasure to having you here today in this webinar. And again, my name is Angela Zhou from CAS. CAS is an organization specialized in scientific information solutions. We're here to help you. And don't forget to explore CAS's website on COVID resources. Thank you so much. And I'm going to turn my microphone to Mike.
All right, well, I'd also like to thank everyone for joining us today. And for all of you in the audience, I just want to let you know about a few webinars we have coming up that you might be interested in. And a reminder, they are free to register for. You can join us tomorrow for our first drug design webinar of the year as we discuss the treatment of cancer medications that target the KRAS mutagen cancers, which has been labeled previously as undruggable. On Wednesday, March 3rd, discover how to protect your technology and IP interests by making sure the appropriate agreements are in place at the start, so you cover essential aspects like confidentiality and proprietary information, trade secrets, inventions, and patent applications. Then on March 10th, join leaders from the Organization for the Prohibition of Chemical Weapons, the National Academies of Science, Engineering, and Medicine, and the Malta Conferences Foundation as they discuss how diplomacy can be promoted through science and share examples of what has been done in the past. We would love to hear what you think about our program. You can reach out to us through the ACS social media channels on Facebook, Twitter, LinkedIn, or by email at acswebinars@acs.org. We're always curious to hear what you think of our broadcasts, and you can, a short survey will pop up directly after the webinar that will take just a minute to complete. Also, remember that an invitation email to view the recording from today's presentation will be sent to all registrants. It will be open for a 24-hour period, after which it will be removed, edited, and posted open access to the ACS Webinars library. And that wraps up our presentation for today. On behalf of all of us at ACS and CAS, thank you so much for joining us today, and we'll see you next time.